Good afternoon. My name is Munnich, and I first want to thank the organizers for allowing me to speak because I'm very excited to talk to you about the novel method our group developed to enrich for Hodgkin and Reed-Sternberg cells using size-based microfiltration. I guess I don't know how to do this. There we go. Sorry. Okay, so let's first talk about these HRS cells. So these cells are the hallmark tumor cell in Hodgkin lymphoma. They are multinucleated, and they are very large. They range in size from 20 to 100 microns, and they're very commonly associated with a lot of T cell aggregates. They're also very fragile, and they're commonly identified using CD30. They also only represent between 0.1 and 10% of the tumor tissue. So all of these characteristics make these cells very challenging to isolate at a high enough yield to do a lot of clinical assays and sequencing experiments very feasibly and also reproducibly. So we sought out this size-based microfilter technology to address some of these challenges. So this technology was originally created to enrich for circulating tumor cells in the peripheral blood. It works using 40,000 pores that are 8 micron in size, and that allows the small cells in the blood to pass through the pores while the larger CTCs are captured on chip. The filter is optimized to then look at these cells captured on chip using immunofluorescence, different molecular analyses, and FISH. So given the very large size of the HRS cells, we asked the question, can we utilize this technology to enrich for the rare HRS cells in the lymph node at a high enough rate to do some of these assays and study these cells in ways that are currently not very possible and feasible. So we started with a cell model. We use these L428 cells, which are a common Hodgkin lymphoma model cell line that are very similar in size to the HRS cells and are CD30 positive. The one major difference we saw with these cells is that not many of them were multinucleated. As background, we used fresh normal lymph tissue, and we made several mixtures of our L428 and lymph node cells, with the L428s making up between 0.1 and 10% of those mixtures. We looked at our mixtures pre and post filtration onto the filter with a few different methods to determine the capture rate we would be able to achieve on the microfilter. So first we looked at immunofluorescence. So we took our mixtures, processed them onto our filters. Those filters were stained with CD30 and imaged, and then analyzed with QPath, which is an open source digital pathology software. So we used a few criteria to identify our L428s, those being CD30 positivity, the appearance of the CD30 stain, cell size, and cell morphology. So as you can see here in panel B, there's quite a few CD30 positive events, and they do look a little bit small, but that's just because the cells are captured in the pore of the microfilter. So sometimes that does make the analysis a little more challenging. But in panel C, we can see a larger L428 captured on the top of the microfilter that's a little more easily identified. So after analyzing all of these filters, we concluded that around 83% of the L428s in our initial mixtures were captured on the microfilter, and nearly all of the lymph node cells were depleted. We then sought to prove we can enrich for the L428s using SNP genotyping. So we selected a few variants we knew to be present in our L428s. And again, we made our mixtures, processed them onto our filters, and then we extracted DNA directly from those cells that are enriched on the microfilter. That DNA was compared with a mixture of cells that were not filtered, and then we also compared that to L428 DNA alone and lymph node DNA alone. And what we saw post-filtration was a reduction of the reference allele signal, and overall our post-filtration mixture is just very similar to our L428 DNA alone. So these two series of experiments told us that we can enrich for the L428s across these various concentrations, and also that we can extract DNA from these cells directly captured on the microfilter, enabling us to do some sequencing. So given these results, we moved forward with a clinical sample. For this, we used lymph node excisional biopsies, and we've been able to assess 2 samples thus far. We did use the immunofluorescence method that we optimized using our cell model for these experiments, and we did look at a few different input cell counts for each of our samples, given that we weren't sure how many HRS cells we might find. So as you can see, starting on the left here, in this like magenta pink color, the CD30 positive cells pre-filtration are a very small percentage of the total cells quantified. But post-filtration, we're now seeing the HRS cells are around 50% or more of the cells captured on the microfilter. This is going to allow us to do a lot of new sequencing and clinical assays that are currently just not as feasible. So even though we're not getting a perfectly pure population of these cells, we're able to get a good yield of these cells. So that is going to enable us to study these cells a lot more thoroughly. You can also appreciate in these representative images the morphology of the HRS cells a little bit better, and you can also see the cells are captured on top of the microfilter, which makes the analysis of the clinical samples much more easy than the cell model. So given these results, our next steps here are to look at a few additional samples using our immunofluorescence method. We're also in the early stages of optimizing some FISH probes to look at the HRS cells that way, and we also want to extract DNA from the HRS cells and do some additional sequencing that has not yet been done on these cells. So if you have any additional questions for me, please see me at my poster or around the conference. Thank you very much.